So good evening everyone, and welcome to this public lecture hosted by the Royal Irish Academy and the Use of the Earth Institute. Just one thing before we get started, this will be live broadcasted, so you might show up on someone's screen somewhere, just to let you know. So I'm Dr. Jens Carlson. I'm working at the Earth Institute, and I should probably start with introducing how I know Cindy van Dover here, where we met. So first of all, she's the world's foremost scientific aquanaut. She's been diving all over the world. And I first met Cindy in North Carolina at Duke University, where I worked as a population geneticist. And Cindy wanted me to get involved in the deep sea world, looking at hydrothermal vents and genetics. And we're still working together on multiple projects ranging from NASA-funded basic research to industry-funded gold mining excursions to the hydrothermal vents. And back then, and she still is, the Harvey W. Smith Professor of Biological Oceanography of the Nicholas School of Environment at Duke University, and she's also head of the Duke Marine Lab. So Cindy was or still is, one of the pioneers in deep sea science and one of the first explorers who discovered hydrothermal vents. And she wrote a book on hydrothermal vents that is now the standard textbook for budding marine biologists. She's published in countless high-impact scientific journals and participated in several TV documentaries. But Cindy's not just a scientist. She's also the only female pilot of the United States Navy's submersible Alvin. And she's been diving with Alvin in a range of ROVs, that's remotely operated vehicles, numerous hydrothermal vents around the globe and other types of deep sea ecosystems. And she's also groundbreaking in using artistic media as means to share her experiences of the deep sea with a non-scientific audience by bringing artists on her research cruises. And she's also runs a traveling art exhibition based on her own and primarily artist impressions of the deep sea. That's a big thing that Cindy's been doing for many years now. And she's also the reason why my lab is still working in the hydrothermal vents and chemosynthetic ecosystems. So without further ado, I'd like to introduce my friend, my mentor, and my colleague, Professor Cindy van Dover.
We have fun together. If you ever get a chance to go out to sea, that's where you really build relationships with people. So we spent, I think, 2 cruises together, a few weeks out at sea, and it's really, really a fun opportunity to talk about science 24/7. So it's really great. So thank you for the honor of inviting me to speak to you all and share experiences. I am giving a very Cindy van Dover, not centric, but it's my perspective, and it's going to be Americentric. I apologize for that, but you'll see some other countries represented, especially Ireland. And so I'm not pretending to give you the whole history of deep sea exploration and exploitation, but just actually, whenever you give a talk, it's a chance to talk about your favorite stuff, right? So that's what you're going to hear, the things I love. Okay, so exploration. I had to put this in because obviously that's what I was going to talk about first, but I need a break when I get to exploitation, so this is my little symmetry. Humans like to explore. We like to go, I need to stand in the right place. Okay, that'll probably work. And since this is from the 1600s, a drawing of an early submersible, I have learned, since putting this talk together, I learned about the remotely operated vehicle Holland 1, and I thought it was named for the Dutch. I thought it was like a Dutch ROV, but it's actually named for Holland, the inventor of submarines, or an inventor of submarines. So, and he's Irish. So that's really fun to learn. So we've been thinking about diving on the seafloor and trying to understand what's there for a long time. Oh, we got into a different, how did it switch? It was set so it would actually look right. Maybe we're just going to cribble along. So this is from the Encyclopedia Britannica from 1823, I think. Through want of instruments, the sea beyond a certain depth has been found unfathomable. And in fact, for a decade or two, we thought the deep sea was a real desert, devoid of life. And so there was this azoic, without life, without animals, this theory from Edward Forbes, a very famous biologist. But he got this wrong. He thought below 300 fathoms, 500 meters, that there was no life. And that was reasonably quickly turned around, in part because of Geo Stars in 1864 dredged up this crinoid from deep. So this was a living fossil. It was the first time crinoids alive had been collected. And so there was a lot of interest in the deep sea all of a sudden, deep ocean. And that led to, in large part, led to the Challenger expedition, where the Challenger, the sailing vessel, scientific sailing vessel, went around the globe. The fun part about the Challenger expedition, I didn't know this till I started looking at some of this stuff. It was the first vessel to discover manganese nodules on the seabed. And so if you think about exploitation, you're going to hear later that manganese nodules are one of the things that's of interest for exploiting for, especially the cobalt that's in those manganese nodules. They're fossil deposits found throughout the world's oceans, actually. The submersible world, I think we really started to get humans down to deep water. In the 1930s, this is the New York Zoological Society. William Beebe wrote a book called Half Mile Down and described his adventures on a tethered ball that just was hung off a crane and dumped into the ocean. Really very primitive. And interestingly enough, he took an artist along. Elsa Bosselman was the artist. She didn't go in the vehicle, but she saw the animals. And this was how he illustrated it, because the camera work just wasn't as good as the artist's perception. So we're starting to get into the deep sea in the 1930s with people. And then this was, what year? I think my crib notes are gone. It's down on the bottom there, I can't see. Anyway, when was the Challenger? I mean, the Trieste dives. You guys aren't going to know. Sometime in the 1900s, after 1930. Well, we went to the deepest part of the ocean, in the Mariana Trench. That was a U.S. Navy-French collaboration. That big star is where they dove off the east of the Philippines. So more than 10,000 meters. And then you might know from James Cameron, the film producer, he built a vehicle called the Deep Challenger and went back again. It's the first time the bottom of the seafloor, the full ocean depth, had been occupied by a human since that Trieste dive. He didn't spend much time down there. There were creaking sounds in the submarine, I understand, but just to let you know, it's a hard place to go. This was part of the X Prize Deep Challenge, but he got there and is making a movie. Then in the U.S., really the submersible activity started to develop through a fellow named Al Vine, who was a geologist. And Al Vine wanted a submersible to go look at seafloor features, the geology of the seafloor, to understand basic Earth processes. And so Alvin was built, and Alvin was named for Al Vine, but also at the time, that's when Alvin and the Chipmunks were popular and just came out. And in fact, do any of you know engineers, or are you engineers? They've got a kind of quirky sense of humor. So Alvin the Chipmunk is involved in the story. And in fact, the mothership for this was called Lulu. That's the name of Al Vine's mother. We have fun. Okay, so Alvin is pretty famous. It's done a lot of cool things, including dive on the Titanic with Bob Ballard. This is back Project Famous, French-American mid-ocean undersea studies, looking at the Mid-Atlantic Ridge, this mountain range that girdles the globe, that bisects the Atlantic Basin. And here's just an example of how technology, for you young kids, this is what we used to do. You dangle a camera and take pictures and then put them together as hard copies on the seafloor of a gymnasium to try to map out what's going on the seafloor and plan the Alvin dives. When Alvin dives, she carries 3 people, and she has, you look at the viewports and you can see about 20 meters out. You can't see, it's like, shining a flashlight on a little bit of seafloor. So you want to plan your dives pretty well. But at the time, if you read this, as with the Apollo project, extensive unmanned surveys are being conducted before men are sent into this region, whose landscape is more alien to human experience than that of the moon. It is not a normal place for human beings. So it was even then a sense of this exploration, spirit of exploration and adventure and discovery. And some really amazing discoveries were made, including one that happened when Alvin sank in 1968. She spent 10 months on the seafloor at 1,500 meters water depth. The people in the submarine, it was launching and the cradle she was in, a chain gave way, and so the submarine slipped into the water. People climbed out, so nobody was killed in that accident. When they brought her up, she had a sandwich inside. They pack a bag lunch for the people in the submarine, and it was edible. Can you see the bite taken out there? So after 10 months on the seafloor, perfectly edible sandwich, which got the microbiologists interested, the deep sea microbiologists. And oh my gosh, I'm sorry. Is there a way to turn this other mic off? I wonder. I didn't look up here. Sorry. So the idea was that, gosh, microbial degradation must be really slow in the deep sea, and it started a whole new kind of business, scientific business of understanding microbial productivity in the deep sea and dynamics and growth rates. Thanks, you guys got it. What's interesting though, do you guys know American food, right? Wonder Bread, do you have Wonder Bread here? Right, preservatives, like you wouldn't believe. Bologna, nitrates, right? It is not going to rot in 10 months in a refrigerator in salt water, right? So it's in brine, it's in seawater. So in fact, you could do the experiment where you put a bologna sandwich and Wonder Bread in your refrigerator for 10 months and it'll be perfectly edible. The microbes just don't grow on preservatives. But it got a lot of deep sea microbiology underway. Very famous scientist, Holger Janisch, made his career on studying the bacteria and how they use substrates. Seafloor mapping, so this is a little bit separate from Alvin, but it gets to where a lot of Alvin's work has been. And it also features a woman. There weren't that many women involved in deep ocean science. But Marie Tharp slaved over these echo soundings. Excuse me. And she would make by hand these maps of the seafloor. And that's how we understood a lot of what the seafloor looked like. This is her 1977 map. and this is kind of, I have this outside my door. I still live by looking at this. We see these seafloor pictures. You want to think about where to go next? Like, look here, Triple Junction, got to study that, right? Geologist. Or this big fracture zone. Or there's a little spreading center up here, the Cayman Rise. Or you go into the Arctic and the Arctic ridges, places we haven't been, the Scotia Arc that the Brits studied recently. Excuse me. I'm so excited, I'm losing my voice. So this is what we had for maps. Now we do amazingly way better maps. But this is what we were basically working with, artists drawing in 1977. Then hydrothermal vents were discovered. 1979 were the black smokers here. Vents are hot springs on the seafloor. So this particular one is a couple of 1,000 meters down. And it's kind of like geysers on land, except it's not intermittent. But seawater percolates down through the ocean crust, reaches hot rock, reacts with that rock. Two ingredients, seawater and rock, and you get this crazy chemical reactions. And the fluid comes out chemically modified, and turns out it's going to be useful for microbes. So microbiology comes into the story again. So if you haven't seen a black smoker, here are these Cayman hydrothermal vents. This is at 5,000 meters. This is the deepest known hot spring on the seafloor. That fluid is coming out right there at that orifice at 400 degrees Celsius. It is so hot that it glows. We can't see with our eyes, but it glows. It's in the near infrared. And then this stuff that looks like smoke is, it's not smoke, it's finely divided metal precipitates. So, and then there's shrimp. So and the shrimp are really cool. They're a whole story by themselves, but I'm not going to tell it. So all that material coming out of the chimney and the precipitate, those chimneys that form, in some places they can accumulate over time to form these massive seafloor, massive sulfide deposits. Those massive sulfide deposits are rich in iron, copper, zinc. They have gold and silver sometimes for bycatch. And with the discovery of hydrothermal vents, suddenly we knew why copper mines in Cyprus, how they were formed. They come from the seafloors. Cyprus ophiolites, this copper mine in Cyprus, is seafloor that was a hydrothermal vent, fossil sulfide deposit, lifted up on land and mined. So it kind of changed our way of thinking about ore geology. Very, 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 very fascinating. But here's the cool part. I'm a biologist, right? So in cooler temperatures, the vent systems are leaky, and you can have diffuse flow coming out that's not, no animal lives at 400 degrees or 350 degrees Celsius. but they will live at this temperature, which is maybe 12, 15, 20 degrees Celsius. So these are tube worms on the seafloor. Who's heard of tube worms? Okay, we got some tube worm people. Who loves tube worms? I find not everyone, it just shocks me, but not everyone really loves tube worms. They're extraordinary creatures. They occur on this particular, especially this really large species, occurs on the mountain range that's to the west of the U.S. coast in Mexico, called the East Pacific Rise. But I've got some video of these guys. They are blood red. You see them duck down into the tube. So they live in a tube that's maybe an inch in diameter. They can be as tall as me. The red plume is exposed and ducked down. They never show the rest of their body that's down in the tube. There's two species in that picture. Okay, this video isn't mine. It cuts over to, this is to remind us, though, that it's a volcanic system. Hot springs are associated with hot rock and volcanism. This is an eruption on the seafloor. So this happens intermittently. These hydrothermal vent systems are ephemeral. They will eventually shut down. Sometimes fluid is coming out for decades, sometimes for thousands, hundreds of thousands of years. Sometimes they get overrun by lava. Okay, back to tube worms. So the tube worms, this plume, so the tube worms, they have no mouth, they have no digestive system. The plumes have hemoglobin, there's blood, and the animal that lives, that's the rest of the animal that's in the tube, is basically a sac to house an organ called a trophisome filled with bacteria. The bacteria are taking the chemicals coming up from the vent. You see that water going upwards? That's not normal. Normally water goes horizontally. This has got a buoyant flux to it because it's slightly warm. It's coming out into a cold seafloor. So the normal temperature is 2 degrees C. So these guys are bathed in warm water. And they have bacteria. So they have bacteria in the trophisome. The plumes, these animals are extraordinary. They are taking up hydrogen sulfide from the vent fluid that's coming out, that chemically modified fluid. So we're sucking up hydrogen sulfide. Hydrogen sulfide is a potent toxin to me and you. It poisons our cytochrome C oxidase system, the thing that makes our ATP. But the microbes that live in the tube worms can take that sulfide and oxidize it and turn it into ATP. So instead of killing the ATP system, it actually feeds the system and the microbes. It doesn't poison the tube worm because it gets trapped, locked onto a hemoglobin molecule, just zaps right onto it and gets carried down to those bacteria. That's why the blood is so red. It's got this hemoglobin. That hemoglobin especially has a binding site for oxygen separate from a binding site for the sulfide, so there's not competition for binding. So these guys are taking up hydrogen sulfide, which normally would kill us, but they detoxify the sulfide. They are taking up carbon dioxide, CO2. They need a carbon source to turn into new cells. The bacteria do this. So the CO2, the carbon, is taken up. We normally respire. These guys are sucking up CO2. Lovely animals. And then, instead of oxygen. So every animal has to have oxygen, right? So tube worm has oxygen, but it needs a whole lot of oxygen because it's got those bacteria that are churning away, oxidizing the sulfide. So they're really exquisitely designed to live in this environment. Tremendous amount to learn from these animals still. They're really just, you gotta love. Now who loves tube worms? Okay, so this is really Americentric because there are vehicles all over the world now that can get you access to the seafloor. So I'm going to tell you about what I know, which is Alvin. So Alvin was built in, like you saw, was built in the 60s, was commissioned in 1964. So it's now just had its 50th birthday. Over the years, every 3 years it gets overhauled and rebuilt. And it just went through major recreation this past, decade. It took a decade. I was on a committee, an oversight committee. It was a decade of being on a committee. Oh my God. So I met twice a week. So here's the new Alvin. And this is what you'll see. Jens was out with us diving in this vehicle. Inside, it's kind of like a jet airplane cockpit. It's got lots of toggle switches and buttons and screens and things. Great fun to drive. I can't drive this new one though. It's changed so much. I'd have to study for a long time to learn how to drive it. And this is just, it has not, I don't know that it has much to do with anything, but it's just so cool how they built this sphere. So people, the reason we can survive at great depths, this will go down to 4,500 meters right now. The reason we survive is because you're in a personnel sphere made of titanium. And here is the titanium for this new sphere being made. This was a technology that was lost in the U.S., maybe in the world, but I know in the U.S. we didn't have it. So I had to recreate it. We couldn't go to Russia to have it done or wherever somebody else might have done it. It's the U.S. National Science Foundation. We had to do it. Everything here is special. This is a one-off. So all the die, the equipment that you're seeing to handle it was all made just to do this one project. In the end, the vehicle, I think, the complete project was $41 million to build this submarine. It's because it's human occupied that it's so expensive. So we can get access to the seafloor without it costing so much. But so this is a titanium. This is half of the hemisphere that's going to make up the sphere. And they paddle it down and they shape it, and they're trying to get it flat enough that they can press it. Isn't that extraordinary? The precision, the tolerances once this thing was pressed and then cut were so extreme. Because any flaw would be, it's a hazard. You've got to have this thing actually not just hold the pressure at 4,500 meters, but it has to do it over and over and over again. It's expected to have 4,000 or 5,000 cycle life. So that many dives. And then we're going to just watch a little bit more as they put this in the press to make a hemisphere. So you see the half sphere up at the top and the pit that's going to be pressed into. I would not want to be that forklift driver. What if you missed? Every scrap of titanium that fell on the deck is swept up and saved because the titanium itself is so valuable. It's so simple-minded. You just punch it down and make a cookie. Those thumbprints. This is what you were in. This is going to be in a movie. I'm sure it will have very dramatic sound effects at this point, but I don't have any music to go. It should have some, like, Star Wars something. Mm-hmm. Then the two halves, I won't show you this, but the two halves are put together with an electron beam weld, so they don't actually put any new material. They tack it with some, I forget what you call it, but anyway, they tack it like with solder, and then they do an electron beam, so here it comes out, the hemisphere. Can you imagine how they were holding their breath? Did we get it right? Did we do all the calculations right? Because they couldn't practice. There it is. Sweet. Alvin on the hoof. Okay, you get the idea. All right. So we do use other things, not just human-occupied vehicles. This is a remotely operated vehicle. It has actually an eye in the sky. It decouples the motion of the ship from the vehicle on the seafloor, so we also put a camera on it. So this is a little one called Hercules, looking on the seafloor, exploring and transmitting video back. Here's the one we use in the U.S. a lot. We used, Jens was out on a Jason cruise. Going over the side. So these are much smaller than typically smaller than the human-occupied vehicles, formerly known as manned vehicles. So off it goes into the water on an adventure. Very fun to watch these things go in the water. It's exciting. You never know what you're going to find. And then from an ROV, all that video feed comes up to a very comfortable control van or control room with lots and lots and lots of screens. This in the front here, this is the pilot, very laid back. He's got a joystick here, and he just is listening to the scientist here. It says, would you go here, go left, go right? So the scientist's role is just to talk the pilots through the work. There's a navigator back here, and then behind the pilot is another row of scientists. And then you can put the video screen around the ship so everybody can watch. So it's pretty great fun to do. And You know, the fact is, I actually would prefer to use an ROV than a human-occupied vehicle because this is very democratic. Everybody can get in that room and talk about the science and see it. There's no secrets. There's nobody who saw something that nobody else did. And even Alvin, it takes lots of video, but you always get the same view that the scientists saw. So this is my preferred way to do science. Jens might disagree. But then I've been down on the seafloor like almost 100 times, and he's only been once. I did want to make sure you all knew that there's some really cool work being done in the deep sea. The Celtic Explorer has been working on the Mid-Atlantic Ridge, studying the Moytira, actually discovering the Moytira vent field. So here's the Holland one that we talked about earlier. So it's actually, this site is really important to us. We think about the biogeography of vent faunas. The fact is you can go to hydrothermal vents in the eastern Pacific and the fauna will be different there from what you find at hydrothermal vents in the western Pacific, different from the South Atlantic, different from the north of Iceland. So we go, and in the Indian Ocean. So we're going around trying to understand who's where and then why they're there, why species are in a certain place, and also about how far a species range is and what their evolutionary dispersal as well as ecological dispersal is. So this Moytira site was the first site north of the Azores on the Mid-Atlantic Ridge. We knew south of the Azores where it was deep and then it shallows up, and we were curious about whether the Azores would be an isolating mechanism for the things that were back on the other side. And so this is helping us get the answer. I don't quite know what the answer is, but Jens does. I think it's different, right? Some ways different. It's a secret. Okay. And then the other thing is I just was seeing this today, the real map of Ireland. I like the way that's spoken because it reminds you all, if you see this map, that it's not just your land base, you've got all this beautiful deep ocean. And in fact, I was talking with Patrick Collins about this porcupine sea bite and the canyons here, and the fact that there's probably really cool things, not hot springs, but what we call cold seeps that we find on continental margins, especially where there's been a lot of organic material deposited over eons. So there's lots of potential for discovery here. Okay, this is a vehicle that I have come to love very, very much. This is an autonomous vehicle. She's on a little cable here for a launch or recovery, I don't know which. And we do when she has if she's on a mission on a cruise, she starts to get a personality as she becomes successful. If she fails, like it took her 4 or 5 days to get her to work right on the cruise we were just on this summer. She's carrying a saddle pack that's a pump to sample plankton. She goes down autonomously. She's programmed. She runs a mission. You can modify the mission by sending an acoustic command down every once in a while to get a little bit of feedback of what she's doing. And then she can go down for 24 hours on her battery charge and just run lines and do adaptive sampling, all kinds of cool things. Very, very great mapping tool. And we've had a chance in the U.S. I don't know if you're doing it here or in Europe, but we're doing European Union. Are you guys part of Europe? But you're not part of the European Union.
We have both.
Both, okay. Whatever. I'm sorry, forgive me. I didn't mean whatever, but I just... I don't know if you guys are doing this, but we are starting to do what we call telepresence. And so here with Sentry, this is Dana Yerger, a really brilliant engineer, who said it's doing a Sentry cruise by telepresence where you throw the vehicle over, there's no video coming back up. You know, for the person doing it on the ship, it's like watching paint dry because you can track the navigation if you really aren't watching much. For people ashore, it's like watching someone watch paint dry. So it seemed like it would be a really boring cruise, but we got all these kids together, graduate students and undergraduates, in Rhode Island. We're the entire science team and the lead engineer were on land, and the ship was at sea. So this is live feed of the vehicle landing on the deck. They sent all the data to us every morning. We would analyze it. The students would just go to work. We'd help them understand what was going on. And then we'd say, okay, here's the mission we want you to do the next night based on what we just learned. And we would pipe that over to the vehicle and then send it down. We'd go back to the hotel and go to sleep. It would go do its business. We would come back to the center the next morning, get new data, crunch it. It was fabulous, fabulous experience. So you can do science from shore on a ship. It's really, really very exciting way to do things. And we were just out again this summer, this time with an ROV, which is a lot more exciting. You see this is what the screen, you can watch it in your home on your computer. This is a Nautilus Live website. There's nothing going on right now, but when it's an active mission. You'll see the video feed. There'll be somebody talking all the time. Here we are looking at the big screen on shore, and we were in these at the controls there doing science again. This time with an ROV. So it's really, really, really very capable way of doing science. And it means we can work internationally. Jens or any of you could get online here. We can talk on, we have chat rooms where we can talk to each other, and it's very fast communication. And Jens could say, pick up that shrimp, and the ship would go, the vessel, vehicle would go pick it up for him. This is by way of introducing how we're working together, a little bit of it. And it's really one, right now, Jens mentioned the NASA project, but that also has kind of been rolled into the SEAP connectivity project, because we're working all in this Western Atlantic, Caribbean, Gulf of Mexico area. And this concept is what we call a seascape approach. It's not novel to us. It's been done in shallow water, but we were applying it to the deep sea, where you take your physical oceanographic information, you understand where the currents go. So we're working from Barbados into the Caribbean, Cayman Rise off of Grand Cayman, into the Gulf of Mexico, and then SEAP sites up here. And we look at where those currents go, and think about where the larvae could go of these little island-like communities on the seafloor. We look at the larval ecology, so we understand where the larvae are in the water column and when. And then we look at the genetics, and this is where Jens's lab and mine work together a lot. So we have as partners, we have in the US, NC State is doing the physical oceanography, University of Oregon is looking at the larval ecology, and then this Duke, UCD, and University of Louisiana team are thinking about the population genetics. We haven't finished our work, but we still have another field season to go next year. We'll be back out to sea with Alvin and sampling again. So just to summarize where our study sites are, we've been in Barbados, a couple of sites there, new site at Kikumjeni that we just sampled this summer, the Cayman sites, Gulf of Mexico, and then on up here. And the question, there's lots of other islands of these habitats in between. This isn't all of them. But we're really interested in a mussel species that's found in Barbados. Is it genetically identical to the mussel species we find in Cayman and Gulf of Mexico? We look at really closely at the genetics and at high resolution, high genetic resolution, to look at where the populations, one population begins and another ends. And also the directionality of gene flow, and also the phylogeographic history. We do this with a number of species so that we can get a sense of what the community is doing. So that's the game we're trying to play. It's very exciting. It's the most ambitious program like this for SEAPs, I think, in the world. SEAPs, I keep talking about SEAPs, so these are not vents. These are along continental margins. Like I said, you undoubtedly have SEAPs on your margin that just haven't been found yet. One way to find them is by looking for bubbles. They have bubble streams. This is really cool. It's just a paper out today, or this week, about orthogenic carbonates. This is carbonate produced at the SEAP by microbes. It captures carbon. There are methanotrophs in there that capture carbon. Really, probably really very important for the carbon budget of the ocean and draw down carbon. And they have these lush communities of invertebrates. The mussels have symbionts inside of their gills, so they're doing the same kind of thing the tubercs are doing. Bubble plumes. So you can find the bubble plumes by mapping. So if anybody's here, a geologist uses the ships to do multi-beam mapping, it's acoustic mapping of the seafloor. Normally you throw away all the data that's in the water column and just look at the data coming from the seabed, because that's what you want is the morphology of the seabed, you really exquisite maps. But if you don't throw away that water column data and look at it with the proper filters, and I can't do it, but my colleagues can, you can see these bubble plumes. And those bubble plumes, we had 2 where we had known seeps, and then we found 3 other seeps close by. And we've now, colleagues of mine have done this along the Atlantic seaboard of the US and found 600 seeps just along our Atlantic margin. So they're kind of everywhere, but they differ in character, so they're not all the same. Seeps aren't all equal. So we have a lot of work to parse out why they're there, how they work, and who's there, the biology of the systems. Great fun. There's so much. There's so much to do, and there's so few people doing it that anybody can become a world's expert very quickly. The tools we use for SEAPSEA, we do mock nest tows. That's how we find out where the larvae are in the water column. That's Craig Young out of University of Oregon. We use Sentry to map, but also to do plankton tows. This pump didn't work. Plankton pump didn't work. It didn't do enough volume, so we didn't get larvae. So now we're designing nets to put on the side of Sentry. And what's nice about Sentry is it can go a meter above the bottom. If you don't tow a net a meter above the bottom, you'll lose it because it'll just snag on something. You just can't control it that well. But Sentry has very precise navigation, so we can get a meter above the bottom. And that's where the larvae are for these seeps and vent systems. We use CTD rosette, so we can put water in these bottles, but we're more interested in the water physical properties, so temperature and density, salinity. And so that's what physical oceanographers use. They also are using moorings and they make regional ocean models to tell us where the currents are going. Like I think I said, we use the ROV Jason, and we use Alvin. And here is Jens coming out of Alvin after his first dive. And you get a happy greeting when you come out, a little bit of indoctrination. Was that cold? Ice water. It used to be nastier than that, but we're now very civilized. Kind, gentle. You know, some people sit down. Everybody knows it's coming, and it's just take it with good grace. You're a good sport. And then, what do you do with samples when they come up? This is us in the lab, a time-lapse video that one of the artists on board took for us. Jens is there in a white shirt. Every once in a while, he pops up there over somewhere around there. But what we're doing is dissecting all the animals, getting little pieces of tissue to be brought back to the lab. It takes a lot of individuals to do the genetic work at the resolution we want. And so we're there into the night. The sub comes up, say, or the vehicle comes up at, say, 5 p.m. You have dinner, you get your samples off the vehicle, and then you spend 10 hours processing the animals. And then finally you fall asleep and come back and process some more. You like to get them fresh and into preservative right away. So it's a lot of dissecting, a lot of sorting, and logging, all that stuff. Okay, good enough. Yeah, we keep going. Okay, so then what's going to happen with this kind of data? Here's a glimpse of the kinds of things we're thinking about. This is work again by Craig Young and Roy He. We don't have the genetic data to go with it, but it helps us think about our genetic data. So here's a tube worm that lives in the Gulf of Mexico. This is the Gulf of Mexico, that's Louisiana. And this species has a 21-day planktonic larval duration. So it's in the water column as a larva for 21 days where it disperses. And we have two cases here, if it's dispersing at 100 meters and here dispersing at 500 meters. And what you see for this very short-lived larval stage, it doesn't really disperse very far, which suggests that if you had this species somewhere else, like over on the other side of the Florida Peninsula, it might be isolated. There might have been an isolating mechanism because maybe it can't get out. So we can test that using the genetic tools. And then if we look at the mussel, this is actually the mussel we get in lots of our sites, seep sites. Here it's just looking in the Gulf of Mexico and out. This was seeded in the Gulf of Mexico. So it has a almost 400-day planktonic larval duration, really, really long. And if it's at 100 meters, it can go all the way up in the Gulf Stream and populate the seep sites up here off New England. If it's shorter, if it's deeper, it can't quite get out so far. A few get out. But it doesn't take that many to get genetic mixing. So these are the kinds of ways we think about where larvae could go, and then we use the genetics to see if the hypotheses can be validated with the genetic tools. Certainly tells us what to expect for directionality in terms of gene flow. Okay, so I wanted to talk about exploitation. When I started, perspective helps because I started doing this in the early 80s when vents were first discovered. And we did say, oh yeah, ophiolites, one day we'll be able to exploit the metals from the seafloor. It was something, a little tagline you put in your broader impacts that we're required to do for the National Science Foundation to get funding. But I never, ever believed it would happen. Just did not believe it would happen. It hasn't happened yet, either. But I think we're getting much closer. So we're thinking now about exploitation. The other thing I want to mention is, right now I'm going to just talk about non-living seabed resources, basically the minerals. There's also exploitation of genetic resources, exploitation of fish. And fish, especially bottom trawling, is really bad, but I'm not going to talk about that. This is my little world, non-living seabed resources. And it's fun to go back to Jules Verne, who also thought in the ocean depths there are mines of zinc, iron, silver, and gold, which would be quite easy to exploit if you're a science fiction writer. So that's kind of a nice legacy. So the kinds of ores we're thinking about, manganese nodules, I mentioned them already. These are the potatoes on the seafloor. They're just lying there on the sediment, all over the ocean floor, but especially off of kind of equatorial waters off of Mexico, stretching out, Central America, stretching out west. And they are related to hydrothermal activity. But there they sit. They take millennia to form. Each one of those little potatoes took 1 million years. So when you take them up, if you rake them up, you're going to get lots of good cobalt. You're not going to have any of those back on the seafloor. So to the extent that there is an ecosystem specialized to live in a manganese nodule bed, you will disturb that probably for a very, 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 very long time. Is that bad? I don't know. And by the way, I'm not an advocate for mining. I'm not an advocate for not mining. I'm trying to walk that line professionally. Personally, I have strong opinions, but professionally, they're emotional opinions, not, my personal opinions are emotional, not scientific. Seafloor massive sulfides, these are hot springs. They call them SMS deposits. I call them hydrothermal vent deposits. These are also fossil. To be useful for mining, there probably have to be, thousands of years of accumulation. So some sites where the volcanic activity is just so fast that vents get run over and they're not going to be good for mining. There's no deposit there. But other places there are large deposits. And we're still exploring for the big deposits. We don't know where they are. You know how hard it is to find deposits on land? Think about trying to do it in the ocean. And then there's cobalt crusts, and these occur on seamounts. Really hard to imagine how those are going to be mined. And they often host, this doesn't look like a very long-lived coral, but big, massive corals sometimes that are the old-growth forests of the deep sea. And so if you take up a cobalt crust, you're also killing all those old animals. So we have to figure out how to, if it's going to happen, we have to figure out how to do environmental management wisely. And my sense is it's inevitable that sometime in the next century, whether it's next year or within 100 years, we're going to be mining the seafloor. And so we have some interesting things going on with governance. There's both national waters and there's international waters. And the international seabed is all this blue stuff outside of those boundary coastlines, right, the EEZs. International seabed mineral resources are under the regulation of the International Seabed Authority, which came into being in 1994 as part of the Law of the Sea Convention. And they are making up the regulations for mining as we speak, and for environmental management. So if there was ever a window to do wise environmental management in the deep ocean, this is it. So do you see my shoulders sagging? There are very few of us. Patrick's one, Jens helps out, thinking about how do we do this wisely? If it's going to happen, how do we do it wisely so that we don't trash the deep sea environment? And part of the challenge is we just don't understand everything about the deep sea environment. I told you a little bit. We know a lot, but do we know enough? Do we know the critical stuff? So that's what we think about. The Seabed Authority in international waters it's like a gold rush. They're just giving away. It's first come, first serve, get a lease on the seafloor. These are exploration leases they're giving right now, so you can't mine, but they can explore and find the deposits. And so here in the Indian Ocean are Chinese and India claims, and then here are claims in the Atlantic, Russian and French claims. I haven't seen any Irish claims yet. And so to do this, they pay the International Seabed Authority money you have to apply and get approved, and they pay a lease fee, and they have certain requirements of exploration. There's some data sharing and transparency things, but it's all kind of developing. And then they're supposed to mine they have the option to mine after 15 years to get an exploitation license. So far, there is no exploitation license given for any seabed resource in international waters. But some of the leases are about to expire for the manganese nodule beds, and it's going to be interesting to see what the mining regulations look like. In national waters, it's an interesting situation, and we have a case study that Patrick especially worked. He was over in my lab at Duke for a while, worked with me in Papua New Guinea. All these little blocks here, so Papua New Guinea, Australia is like way down there. This is Manus Basin. It's a volcanic back arc spreading center. So in that ocean basin, there are hydrothermal vents, hot springs. And all these blocks, this is from 2010, so it's probably different now, but all these blocks, red and yellow, are leases or leases applied for exploration by a company called Nautilus Minerals. They're the first mover for deep sea mining. And I got to know them back in 2005. They asked me to help out. I was immediately, when my colleagues found out I was doing this, I got slammed for being a greenwasher, right? That's why they came to me, because I knew about deep sea vents. But they came to me in such a way that was like, look, we think we can do this. We'd like to do it well. Will you please give us your advice? And they're thinking about the environment. How do you help them think about the environment in Hot Springs? And so I said, yes, I'll share my knowledge. I can't I feel I can as an academic scientist hold back and not share. So I've been working with them since 2005 as a research contractor, not as a consultant. So I do research. I don't do anything. I go to meetings and talk with them, express my opinions. So here's why they're going to these seafloor massive sulfide hydrothermal vent ore deposits. Here's land-based ores: gold, zinc, copper, and kind of the typical land-based quality now. The good stuff's all gone, apparently. And then on the seafloor. And so if you compare to land to seafloor, you can see that especially in copper reserves in Saguaro 1 and Saguaro 8, two of these vent fields, are really quite good. And copper was the thing everybody's after. China, explosive development in China, India, copper's in high demand. So commodity prices rose before the big economic downturn. And so there was a lot of excitement about going after this. It was economically feasible. Then the downturn, and they slowed down, and then they picked up again this company, Nautilus Minerals, to try to develop the mines and actually start mining, extracting ore. They have permission, they have the license from the government of Papua New Guinea to do this. They did have a litigation with the country over some funding, but that apparently has been resolved, and so they're back on track to get in and mine. Still don't know when. It's always like next year. So hopefully we'll just continue to be next year. But anyway, the ore quality in these hydrothermal vent systems is quite good. Here's how they want to do it. There's several different ways of doing it. Nautilus has chosen a very complex way. I've seen some much simpler designs. But they put vehicles down on the seafloor, these mining tools. Here's shows you how massive they are. Here's a person, that little guy there. They're massive tools. They're designed after what's used in coal mining, I think, maybe diamond mines as well. And it's basically open cut mining on the seafloor. And it's kind of out of sight, out of mind. It's great. They show there's nothing living down there, right, in that picture. But in fact, they're at hydrothermal vents with these animals. So here's a hairy snail and a black snail. These snails have symbionts inside their gills, so they are doing chemosynthesis, chemosynthetic community. These mussels, again, have bacteria inside their gills. So there actually are some really beautiful animal colonies there. And so the mining company, Nautilus Minerals, wants to protect these animals. They're likely to come back fast. These systems are, the animals are adapted to failing chimneys. The chimneys fall down, so they lose their habitat. They have to recruit back. So there's a lot of larvae in the water column. A single mining event may really have no lasting environmental impact. That the animals could come back within a decade, maybe. We don't know, but the potential seems there. In my opinion, I think that's a reasonable estimate. The problem is cumulative impacts, which is what you're going to see in another click away. Sorry, this has gotten out of wrong resolution. But so I was thinking about what some of the physicochemical impacts would be, what causes of impacts on the environment. Loss of habitat, because they're going to just pull up that ore. Degradation of habitat quality. Who knows? They could change the fluid will still come out from the vents, I think, but they'll change that. Could change that equilibrium condition chemistry. So the animals might not be able to tolerate it for at least a while. Fluid flux regimes, maybe they'll slow down, maybe they'll go faster. It's going to be sediment plume, noise. This is thinking just strictly on the seafloor, not what happens with marine mammals or anything else, but seabed effects. Maybe plumes from return water will be harmful. And then the biological consequences of elimination or reduction of local populations. By definition, you'll have decreased reproductive output because you took away the animals, loss of larvae and zooplankton in the riser system, because when the ore comes up, it gets dewatered on the surface. All those things get filtered, killed. I don't know if that matters, but you're going to lose them. There's a potential for local extinction of rare species, perhaps. Regional, global is less likely on a single event. Decreased seafloor primary production, by definition, just because you've disturbed the system, probably comes back pretty fast. Decreased diversity, again, by definition, because you took all the animals away. Mortality, impairment due to toxic sediments, maybe. I don't know, altered behaviors, maybe. But then, what if you just take all those sites in Manus Basin? Then you're in trouble, right? So it's the cumulative impacts. If you take them all away really fast, that's bad. Maybe you could take them away, do one site here, decade 1, and then 2 decades later you go back and do another site. Wouldn't that be wise? That's probably not how it's going to happen. But you can imagine some mitigation tools that you could apply, management tools. And so that's our job. Patrick, me, Jens, we all think about how do you mitigate the impact? How do you help do this wisely if it's going to happen? And then this is just from the Nautilus website of what they envision all this stuff to look like. And if you've got the patience for it, it's kind of fun to watch. I know I'm going a little bit long. I don't think I have too much more after this. So they have all these. So it's a special built ship, and they have a tremendous number of mining tools. The things that go, assets that go in the seawater. My experience with assets that go in the seawater is that they really are difficult to keep operating. Right, so there's a big expense here, and a big, I'm not the engineer, but it takes a lot to keep them going. Cables fail, grounds. These are a lot of mechanical stuff going on in these vehicles. It'd be really interesting to see how they fare once they get in the water. It's a good experiment, really, and it's got to be advancing deep sea technology. So from an engineering perspective, this thing goes down the seafloor. One of the things, Nautilus Minerals is actually very forward-looking in this environmental management. One of the things that they're willing to do, and Patrick is the one. You guys all know Patrick. I keep talking about him over here. He's the one. He and I were at a workshop with Nautilus Minerals, and they said, "So, what do we do?" Because I was starting to talk about restoring these sites, like on land. Mining companies have to restore, rehabilitate. What are you guys going to do? I said, "Well, tell us. What should we do? I mean, how can you not want to work with somebody who says, what should we do?" So they're going to, I think they have mining plans where they would avoid the high density biomass. That's a good strategy as long as you keep the plumes away from it as well. They may leave them there indefinitely. They have a reserve site upstream that they will not mine. Well, this one is being mined. That could serve as a source of propagules to seed this site. And they are willing, this is happening, they'll have ROVs in the water watching, and they can go off and do work. So they can go in front of the mining tool and pick up animals and move them to behind the mining tool at basically no cost, because they're just pilots looking for, practice, so they can move them. So they've started to build little things to facilitate recovery, to try to move it along a little faster. So very, very clever things that they're thinking about, and they get criticized a lot, but they are trying their best, and any really good suggestion and even some bad suggestions they'll consider and try to make happen. But yeah, I wish I'd put some animals in these pictures, because that's not what the seafloor looks like. So they have these two different mining tools. One's a bolt cutter, one's a auxiliary cutter. Their idea is to move all the stuff into these little sheds, storage sheds on the seafloor as ground up stuff, and then to lift it up to the ship after grinding it. So they collect it and then they move it. So it's very, very complicated. It's something to watch. If this happens, watch your news and watch the websites and see if it really works. It's pretty phenomenal. It's good practice for thinking about mining on the moon or Europa. Okay, you get the idea. Pretty crazy. Expensive, very expensive. They are backed by big gold mining companies, Barrick Gold, Technico, those kinds of things have put in hundreds of millions of dollars to make this happen. So here's a part of it's not my problem, but what concerns me is that out of sight, out of mind, how many of you from one day to the next think about the deep sea and the animals, beautiful animals that live there? Today, maybe. I don't know why I don't care about the bottom of the ocean, but I don't. And this is what I think a lot of us feel we're up against. It. Why should you care? And it's difficult for scientists to articulate. You probably want to hear about numbers. So tell me about the carbon that gets captured by these systems, and tell me about why I should care about those two words and how that has been translated into dollars in biotechnology or euros in biotechnology. And that's especially for biologists like me, that's hard to articulate. But we're working on it. We're going to get there. Oh, it says that actually says keep your hands off my bottom, hands off our bottom. Okay, so it's Bloom is Claire Nubian is a photographer. She put together a really beautiful book of photos of the deep sea, and she has this non-profit to try to protect the seafloor. And one of the ways they're doing this just came out in the Guardian this past month, and she's working with Lush Cosmetics, and they're doing body paintings of deep sea animals. I just picked two out here. The animals really are beautiful. So this is her way of trying to get the general public to care about the deep ocean. And Jens mentioned I take artists out to sea. In this last cruise, I took Jim Toomey, who's an American artist, happens to be a Duke alum. And he's got this guy, Sherman, who's a shark, a very intelligent shark, but really lazy. And then the turtle is Fillmore, and he's bookish, pompous, principled, sensitive, and right most of the time. And then there's Ernest, who's like this brilliant brain surgeon, and he just keeps wanting to do the next evil thing. Oh dear, that's too bad. And I get so small, I can't see it here. Ay. Well, so we were in the Gulf of Mexico. There's something called the Sigsbee Deep. So Sherman is saying, oh no, I guess it's Ernest is saying, we're going to a specific part of the Gulf of Mexico called the Sigsbee Deep. And Sherman says, what's so special about it? It's 12,000 feet deep, 300 miles long. Sherman's thinking about it. Ernest is thinking about it. And Sherman says, so, like, it's just a big hole, huh? And Ernest says, oh, if you want to suck the grandeur out of it, yes. Anyway, sorry it's not as well presented as it should have been, but you get the idea. So Jim Toomey is going to do he also does videos, cartoon videos where he might be speaking or there's real video, and then he has the cartoon animals come in and out of it. So he's going to help us do some outreach and try to get people to think about what's on the seafloor. So we work on it in different ways. And so my final words is going to be really tricky because who knows what I said. See if all the words are there. In the deep sea there live strange creatures of extraordinary beauty, exquisitely adapted to extreme environments, like I hope I conveyed to you with the giant tube worms. And there exist deep seabed resources useful to society, right? I'm not saying we can't go there and not take it, but we have to understand it. We may choose to disturb these systems to gain economic or often it's strategic advantage. Countries that don't have these ores just need to know how to get them. But we must make these choices with attention to what we might lose as well as what we might gain. That's my real message. We need to know. We need to know a lot more. And then acknowledgements, funding from that. I wanted to make sure you knew I had funding from Nautilus Minerals, the mining company, as well as the National Science Foundation. Many scientific colleagues, including some who are here, students and postdocs in my lab, and lots. When you go to sea, you have just a whole army of people just from all different places helping make things happen. The shipboard crews that they've produced. And then I stole pictures off the internet. That's it. Thank you very much. So I'm happy to answer questions. All right? Yes?
Just, sorry, could I ask about the mining? Is it actually going on? Or was that just animation?
It is not yet going on. There's no deep ocean mining going on. There's shallow water mining of diamonds, right, real shallow, and phosphates are, I think, being mined in shallow water. But the deep stuff, not yet. So the technology is being built. There's been, I think, test mines maybe in Japan or Korea. But the actual commercial mining, no, you will not find ore from the seafloor in your radio or cell phone just yet. But I think it's debatable. I listen to economic geologists and engineers and technologists and scientists, and sometimes you hear it's going to be tomorrow, and sometimes you hear it's going to be a decade, and sometimes you hear, no, it's not economically reasonable at all. So I don't know what the answer is, when it will happen. I fear it will be sooner rather than later. Stakes are pretty high. In the back, yes.
Hi, Cameron O'Connor from the Earth Institute in UCD. A fascinating talk. Can I ask you about exploitation of biological species? And I suppose a double question. One is, have you looked at because you're looking at genetic diversity, do you see opportunities in what you see in that genetic diversity? And secondly, I had the pleasure of visiting a Spanish company or a biotechnology company who are trying to exploit marine organisms through the study of the organisms, identifying, for example, anti-cancer compounds,
Sure.
and then making them synthetically rather than trying to go down and get more of their furry friends.
Yeah. So ideally, if you're going after genetic resources, it won't be harvest-based, it will be synthetic-based. That's not to say that's how it's always going to happen, but that's, in my mind, I think it's going to have no harm to the environment because you're going to take a few animals and then look at their genetics and build from those gene sequences and what you find and develop the chemicals in the laboratory. But I don't know that that's entirely how it's going to happen. There is no for international waters, there's nobody who has responsibility for the genetic resources. So that's a big concern. A lot of people are thinking about how do we get those right. That maybe the International Seabed Authority should look after them. The genetic, doing screening for therapeutic agents is something pharmaceutical companies do really well, and there's some scientists who have specialized labs for doing that. We don't think about that right now. There might be people in Ireland or elsewhere who'd be interested in that, and there are large collections that could be scanned if they're frozen or in the condition that they could use. There have been products developed from hydrothermal vents and other deep sea environments. Some of the high temperature enzymes for molecular biology come from hot springs. Some from terrestrial hot springs, some from hot springs on the seafloor. There's a lot of interest in cosmetics from deep ocean biochemistry, and so that seems to be a growth area of all things. You could buy lip gloss with something from hydrothermal vent animals, and it probably costs a fortune. So there's opportunity there, and it's not been, a lot of products come from marine environment, but there's a lot of diversity in shallow water. So the deep sea stuff, I think, is still kind of, because it's expensive, you kind of have to tag along with a scientist. One of the things about doing deep sea research is often it's an apprenticeship. It's not obvious how to get out to sea and to the bottom of the ocean unless you've been on a ship and seen somebody else do it. So, so the pharmaceutical companies kind of come to us and ask for help in digging out. In the back?
Yeah, just a triple question for you. First one was the mussels, 500 meters, New Mexico.
Mussels as?
The mussel sites that you were showing us, New Mexico, at 500 meters deep, you said?
Yes.
How did they come up? How did they manage to make it up to 100 meters from that depth?
How do they come up to the surface?
Yeah.
So animals can come up from deep in the ocean. We get them up sometimes from 4,000 meters, they're often dead. But from a couple thousand meters, they come up alive. They don't have gas volumes. And so,
how did they mechanically come up?
Oh, mechanically. Oh, you put them, you collect, we have manipulators on the submersible. For shrimp, we use a slurp gun. We vacuum them up.
No, sorry. You showed us a map, and you said that the 500-meter mussels migrate up the coast of America.
Right.
Yeah. So how do they migrate?
Oh, through their larvae. So that's why we're looking at where the larvae go in the water column. So those trajectories we saw, that was not an adult mussel walking. That was the larvae. They release gametes, so eggs and sperm. They fertilize, form a little larval stage called a villager that can swim, and it gets carried passively by the currents, or they can move up and down vertically, but they can't really move against the current.
The second one then is the manganese nodules, potato size on the bed. They said they took 1 million years to form.
Mm-hmm.
Right, on the bed. Now, obviously, is there any other strata on the Earth that has those manganese modules in geological surface that we can see now? And if so, what are they called, or what's the strata formation that's that?
That's a really good question. I hadn't thought about where are the fossil manganese nodules. I don't know. I don't know that anybody. It seems like I would have heard about that if there were fossil manganese nodules. I don't know. I will ask my geology colleagues. I'm going to go see some next week, and I'll try to find an answer. I don't know how to get it to you, but it's a good question.
And the last one is, when will real estate come down the Ocean Club?
When will real estate? You want to move in down there next to
Mining, yeah, real
next to
estate.
Tuber
When will that happen?
Well, in the international waters, you can go now. Good question. Yeah.
So on land we have classification systems for endangered and rare animal and
Yeah.
plant species. Is there something similar in the deep sea, or is there just not enough information?
There is for some of the deep sea corals. They're on the CITES list as endangered. I don't remember which ones. Do you remember, Patrick? There are some that we can't just go out willy-nilly and sample corals or fish in the U.S. We can't. You have to get special permits. So you can collect them, but you have to have permits. So it's corals are the endangered ones. A couple of species of corals are on the CITES list. And then the rarity is a really interesting question. I have a PhD student who's just starting, who I've asked him to think about this, that when you look at, we measure rarity one way is you look at a species effort curve. So you look at accumulation of species as you sample more and more. And what you normally see is that it goes asymptotic, that you get to a point where you've sampled everything and you pretty much know everything that's there. In the deep sea, it never reaches an asymptote if you're going into the soft sediment environment. Hydrothermal vents are a little different, but in the soft sediment environment. So there's a very large number of rare species. You can keep sampling, you're still going to find a singleton. And so do we care about those rare species? So I was thinking about how do you handle rare species when most species are rare. 90% of the sample is rare species, things that you only find one off. And you have to go, how much more do you have to sample? So do we care about that? How do we value it as a society? And I think you hear one of my messages is trying to get people to think about why these things matter. And it is hard. It's hard to, do you care about that little nematode of which we only have one of? Or do you care about the type location of the species that's going to get destroyed? I mean, Mammoth Basin now is the type location that Solwara one side of many new species. Do we care that it's going to get destroyed? Probably not, but could. So it's the societal, how much do you care as a society? There's some studies now about willingness to pay. How much would you pay to protect an area or to restore an area? I mean, think about restoring, if you're going to mess this up, how are you going to restore it? You can't. So what will you pay to protect it? I don't know. Depends, I want my cell phone, right? You're going to not pay so much to protect that seafloor. I don't know how it's going to come out. What I hope is we can have the cell phone and the biology. Right? That's our job, is to figure out how to do that. Any other questions in the back? Yeah.
Can I ask you one more? Do you think that genetic diversity in the deep ocean can teach us anything about diversity on land and how we will deal in the future with changing environment?
Oh my goodness, that's a very good question. I don't know that I have a really good answer on my feet for that. I'm sure. But I mean, it's a question.
Are you learning now about diversity there that is surprising you, that is saying things to you that you probably wouldn't have predicted, and potentially that could be used? Same question I'm asking.
Probably, the kind of the high order stuff. We get this question all the time. It's like, why do you need to do this in the deep sea? Why don't you just do it on land? And so we have to constantly justify why we would spend the money to study something down the deep sea. I think there are ocean processes. I don't have a good answer. I could come up with one, but I don't have a good example, I guess is what I need to say. Can you think of a good example?
Well, one thing you have down there is it's probably undisturbed. They have undisturbed locations, and that's quite unique compared to terrestrial areas. So if we can study how it would look like when it's undisturbed, we can take lessons from that and move it up to the terrestrial environment. But I also think that we should sort of close this, and we should thank Sandra Van Dover for her talk.
Thank you very much for your
Questions, so we can continue those questions and discussions out at the wine reception. It is in front of the building to the left. But just before that, we at Earth Institute has the gift for Cindy here.
Aww.
So that's from Earth Institute.
Thank you. Is it It is an Earth. Brilliant. Thank you so much, Earth people.
All right, so if you guys want to move out into the wine reception.
Let's do.